Hi everyone, this is Dr. Wang. Today we're going to do this assignment called lab9.2. This is where we analyze the data generated from the FOA assay. All the plate images are provided in this folder right underneath this assignment. Please select the images based on the type of mutant plasmid that your group has. In this image, the plate on the left is the SD plate and the plate on the right is the FOA plate. So there are obviously some differences in growth pattern exhibited by the yeast strain. So we're going to go through the reason to explain what we're seeing on this plate. So first of all, on the SD plate, all three yeast strains are growing. So why is that? The SD plate we're seeing is missing three essential nutrients: histidine, tryptophan, and uracil. And we can see that the genes responsible for the production of those nutrients are missing in the yeast genome, right? So here they're not here in all three yeast strains. However, those essential genes are provided by plasmid that are either carried by the yeast or transformed by us into the yeast. So here's TRIP1, URA3, and HIS3. So here, all three yeast strains are expected to grow on SD plate. So SD plate is also a control plate for this experiment to ensure that we have plated roughly the same amount of yeast on all the plates. So now let's look at the growth behavior on FOA plate. So the difference here is uracil is provided in the FOA plate, and FOA plate, of course, include this chemical FOA. Based on the FOA map that you have already made, you already figured out that a functional URA3 gene in combination with FOA is going to generate a toxin that's going to kill the yeast. So then there's not going to be any growth, right? So if we look at the situation in SP1, we have MSH2 that repairs the URA3 gene to ensure that URA3 gene is functional. So under this condition, the yeast will be killed, so we expect very little growth. And now let's look at SP0. So in this case, URA3 gene is prone to mutation. So when it's mutated, then this is not going to happen. So we do expect a little bit of growth in SP0. So let's see if this is what we are seeing on the plate. SP0 is over here, so we can see that this spot right here has a couple of colonies, and those colonies are the result of mutation in the URA3 gene that allow the yeast to grow in presence of FOA. And if we look at SP1, here there is very little growth as expected. So here SP3, this is the actual data. So here we have a mutated version of MSH2. What we're trying to figure out is whether this mutation in MSH2 affected the function of MSH2, right? So here we are seeing a couple of growths. Right, so the question I have for you is the mutation rate high or low right here? And then what does it mean? Does it mean that the missense mutation in MSH2 is a defective allele or a pseudo wild type allele? And on this FOA plate, we can also see the reason that we plated three different concentrations on the plate. So under OD1 concentration, the density is so high that we barely see any differences among the three yeast strains. And under this concentration, OD 0.01, there's so fewer colonies in this column, it also makes it difficult to compare the growth pattern among the three yeast strains. So here OD 0.1 is the optimal density for us to make this comparison. And the data could vary from group to group, so having a range here will allow you to pick at least one column to allow you to draw conclusions. Coming back to this assignment, lab 9.2, in this assignment, you're supposed to describe the growth pattern for the 3 yeast strains and the 3 different plating concentration in both the SD plate and the FOA plate. So this is the data description part of the results section. And you're also going to explain why the colonies are growing or not growing on those 2 plates. So here, this is the conclusion part of your results section, adding the goal statement and brief mentioning of the method. So here is basically how you write the results section for your lab report. So please, once you're done here, copy paste what you have into your lab report.